biblical text. There it's called the equidistant letter sequences, where every seventh letter spells out something that's relevant to what's going on, and so forth. If you've heard about the Bible codes, it's the exact thing that's going on in your DNA as we go to get the messenger RNA. I won't go through the alphabet issues. Any combination, 3 out of these 4 letters define one of the 20 amino acids plus some punctuation, stop and start bits. And we could go through that. I won't take the time here. We've got more important things to talk about. The code will adapt itself if it's dealing with the power systems, but that's mechanics. What happens if you have a string of these codes? You need some kind of robot to read it and manufacture what it's telling you to manufacture. It looks sort of like a tape recorder. You've got a transfer RNA device that reads it like a recording machine. And as it goes along, it takes the code and builds the protein to get a string, a protein chain that then becomes the robot that you're trying to build from the master copy. And then that chain will fold upon itself because of the chemical and electrical charges into a 3-dimensional machine that is to do some particular function. Let's take one example. Let's take hemoglobin. Your hemoglobin is 574 amino acids long. These 574 amino acids have to be in the right sequence. You have 36 of these are glycine, 68 are alanine. Of the 20 different types, you have a total of 574. Now, it turns out from mathematical switching theory, you can tell what the chances are of getting that right by accident, okay? It's called specificity, and that's the formula. To make a long story short, you have, with 574 elements chosen from an alphabet of 20, you've got 10 with 650 zeros after it as your get it by chance. Now, here's the point. If you get it wrong, that's called hemoglobinopathy. It's fatal. Sickle cell anemia is a derivative of this. In other words, they have to be precisely right. Could that happen by chance? Mathematically impossible. You want 10 to the 650th power? There are only 10 to the 18th seconds in the history of the universe if you accept the 15 billion year history of the universe that the scientists talk about. There are only 10 to the 66th atoms in our entire galaxy. There are only 10 to the 80th particles in our galaxy. 10 to the 650th power. See, in physics, anything that's more rare than 1 chance in 10 to the 50th is defined as absurd. So 10 to the 650th is not anywhere in the realm of having been occasioned by chance. It had to be designed, and designed very skillfully. Now, there's an idea that they were indebted to Michael Behe for the concept of irreducible complexity. How many of you ever handled a mousetrap? It has 5 parts. For years, they've tried to make a better one. It's pretty hard. They have a wooden platform, a hammer, a spring, a holding bar to hold the hammer back, and a catch to hold the holding bar. 5 parts. You have to have all 5 to catch a mouse. If you have 4 of the 5 parts, you don't catch 4/5 as many mice. Okay? That's called nonlinearity. You need all 5. Follow me? That's called irreducible complexity. Let me give you an example in real life. Here's a bacteria. It's a single-celled creature that has a little tail that propels it through fluids, right? Here's a diagram of it. It's a single-celled creature. I want to look at the place that the flagellum joins the cell wall. Here's a microphotograph of it. It's an electric motor. It has D-rings. It has multiple armatures. It has a double wall protection. There are 40 parts to this, any one of which is missing, it doesn't work. It's an example of irreducible complexity in real life. Here's the equivalent electrical diagram of it. Any one part missing, it's busted. Did this happen by chance? Hardly. Very skillful design. The real issue, the reason the Darwinists can't explain the origin of life is because they cannot explain the origin of information. The origin of information. Which came first, the DNA or the proteins? It takes protein to construct DNA, and it takes DNA to construct proteins. Which came first? Come on, guys. Both had to be created at the same time and designed to a consistent system architecture. That same code, those same acids are the ingredients of all life in the universe, which proves it all came from the same design team. All came from the same software house, okay? All right. Psalm 19, we all know the first 4 verses. The heavens declare the glory of God. The firmament showeth his handiwork. Day unto day uttereth speech, and night unto night showeth knowledge. There is no speech nor language where their voice is not heard. Their line has gone out throughout all the earth, and their words to the end of the world. Now, what's interesting about this passage is the information science emphasis. The heavens declare, day utter speech, knowledge, speech, language, words. Information science is the issue that underlies all of this. See, the origin of information can't be explained. The irreducible complexity refutes design by chance or accident. The choreography of the chromosomes defies any random theories. Digital codes demand pre-planning. No system dependent upon subsystems can survive random failures. It takes design, skillful design. Well, let's move on. We've talked about the tissues. Now let's jump into some of the issues. We're tampering with the engines of creation. I want you now to stop and put yourselves in this picture. By the way, anyone that thinks that some of these ethical issues are simple are naive. You have, put yourself in this, you have a 6-year-old daughter or son who has a rare bone marrow disease, anemia, bleeding disorders, severe immune problems, and likely to die of leukemia within the year. Can you picture that? Try to put yourself in those shoes. Your doctor tells you that there's a 90% chance of curing your child through a cell transplant technique from an umbilical cord of a matched sibling, which requires, of course, the use of a screened embryo. What would you do? What would you do? Would you take advantage of that? Of course you would. This is an actual case. Molly Nash was born with Fanconi anemia, destined for leukemia. The only effective treatment known is to get a batch of healthy cells from a perfectly matched sibling to replace the child's faulty bone marrow cells. And since each parent carries both a normal and a faulty version of the Fanconi gene, each pregnancy has a 25% chance of having an affected child subsequently. So what do? They created embryos by standard in vitro fertilization in a laboratory dish, tested for the presence of the disease gene. Only those testing normal are then transferred to a woman's uterus. Only 2 of 15 tested were normal, and only 1 of those 2 was healthy enough for transfer to Lisa Nash's womb, which they did. This designer baby saved the sister. The Colorado couple used genetic tests to create a test tube baby that would have the exact type of cells desperately needed to save their 6-year-old daughter. She now has a 90% chance of being free from the marrow disease. Genetic screening is clearly going to be part of our society, our culture. And that little girl is healthy today. Her baby brother whose umbilical cord was used, he's also doing fine, so the family's doing fine. So this is real life stuff, and it's going to be more and more. See, we're dealing here with the issue of differentiation. There's a very strange mystery that goes on in cytokinesis. If cytokinesis results in identical cells when they split, why do they differentiate? If 2 become 4, and 4 become 8, and 8 becomes 16, and so forth, identically, what's happening when they suddenly start differentiating? Because you know what happens. Pretty soon you get some different ones. There's a dark line goes down the middle. It's the backbone. And as the time goes on, you have, of course, these identical cells differentiate into 210 different types of cells that become the tissues that make up the organs that make up the embryo. Why do they differentiate? And you all have seen these, the development of the embryos. While they're still undifferentiated, they're called stem cells. They use a code of 4 to define the 20 amino acids that make up 100,000 different proteins to make the cell operate. But then for some reason, unknown at the present stage of understanding, they start differentiating. And before they differentiated, they're called stem cells. And there's over 200 different kinds of cells that get differentiated that make up the tissues and organs. There's a total of 10 to the 13th cells in your body, which is a bunch. Now, one of the crafts here is to take a nucleus from some other entity and take this nucleus out of the stem cell, put that nucleus in. It's called a nuclear transfer. That's what we call cloning. And you end up then with a replica of the donor. And that's what cloning is all about, properly called nuclear transfer. And we all know about a clone in sheep's clothing, the famous Dolly sheep in Scotland. A needle-making machine is the key to the whole thing, where they were able to remove the nucleus and insert a nucleus of